Good morning, everyone. My name is Lacey, and I'll get started. I just want to say thank you to Julie and the Alliance for inviting me to speak here today, and also to the families who really over the past 2 years have reached out to me and told me their stories and told me about their kids who have really made the registry possible thus far. So just to kind of give a basic overview, so what is a registry and what's the purpose of really putting one together? So a registry in general just collects information from a group of individuals that really can be accessed for future research, and it just allows researchers to have a patient population to really just tap into. So the purpose of the PCDH19 registry in particular is really to collect both clinical information as well as the specific genetic information from individuals who have PCDH19 related epilepsy, and to put it all into a centralized database so we can have access to the information that's in the database as well as to have access to this patient population if there are additional studies that researchers may be interested in pursuing, and they just say, okay, we really need to find people with this particular type of variant that we want to study. How do we reach out and how do we find people with those particular variants? So we have this population to say, oh, we have a group of people, we can see if these particular families are interested in participating in your study. So as it stands now, our enrollment process, some of you have been through it, some of you are considering going into it. It's a little bit cumbersome, and we're aware it's a little tricky. There are several steps involved as it stands right now. So right now we're asking you guys to read through and sign a very lengthy consent form that outlines the registry and what we're going to be doing with the information. We also ask you guys to sign a records release form that allows us to get a copy of your child's medical record, so we can kind of see a little bit more in depth of your child's seizure history, development history. And we're also asking you guys to fill out a medical and family history questionnaire. So we really want to see things from your perspective. We get a copy of the records to say this is what your neurologist is seeing, but what are you seeing at home? Because there are some nuances that might not be picked up in the clinic that you really think are important for us to know. And I know that there are some registries out there who kind of have an easier process of enrollment. A lot of places kind of do things online, so you just click a couple buttons and you're enrolled in another registry. Ours is a little different. We kind of have this whole curation process where you're signing release forms, but it really allows us to do a thorough data collection from different avenues and to carefully vet the information and include information that's really important and that would be really informative for people to be able to have access to. Okay. So now the important part, right? This is what we really want to know. So where are we right now? So our enrollment updates, we currently have 33 participants enrolled at Boston Children's Hospital, and then at UCSF there are an additional 5. So this is kind of a little table that shows the number of people we've actually been in contact with and are in various stages of the enrollment process and who have completed the enrollment process. So right now at Boston, we have 33 participants who are actually enrolled, but we have another 15 people, which includes 14 probands, or children who have PCDH19 related epilepsy, who haven't quite finished the enrollment process. Our 33 enrolled include 24 probands and 9 unaffected carriers. We're including family members who also carry the PCDH19 variant because we're hoping in the future we're really going to start looking into why some people have symptoms and some don't, and we're thinking more specifically those carrier moms that we haven't quite figured out what's going on. We've heard yesterday a lot about unaffected fathers. We talked a lot about mosaicism and why we don't expect males to be affected, but when we see moms carrying these variants, we're still kind of not really sure why that is. So we're enrolling them even though they have no symptoms, because eventually we might want to be able to look a little bit more into what's going on there. So a little more of those who are enrolled. So of our 24 at Children's in Boston, they're all female right now. We haven't enrolled any males yet. And out of the 24, 13 have de novo variants, meaning that they didn't inherit the variant from either parent. 5 of them, the variants were inherited from mom, 4 inherited from dad, and 2 of the 24 were not sure about inheritance. Mom and dad haven't been tested yet, or there aren't really plans for getting tested at this time. So who are those unaffected carriers that we have right now? So of the 5 participants who have a maternally inherited variant, we have all 5 moms enrolled. We have 2 of the 4 dads enrolled, and then we also have 2 unaffected sisters who are sisters of the people who have maternally inherited variants. So there are a couple females in the family who have the variant and have no symptoms. And kind of looking at the same pattern at UCSF. There are 5 individuals enrolled, 4 probands and 1 unaffected carrier. And looking at the same pattern, 2 of those are the new variants in which the variant was not inherited from mom or dad. One of them inherited the variant from dad, and then there's 1 proband who's awaiting parental testing. And the 1 unaffected carrier is that father there. So we're going to dive a little bit into the clinical information. So we are going to restrict the analysis to the people for whom we have enough clinical information to really be able to tell the story. And we're going to start with the PCDH19 variants themselves. But I was thinking about just diving into talking about what those PCDH19 variants are, but based on some of the questions I heard yesterday and some of the conversations that I've had with parents thus far, I thought it might be helpful to kind of take a step back and go on a little bit of a tangent to make sure we're kind of all on the same page with what do we mean when we talk about a PCDH19 variant, and what exactly does it mean when we talk about mosaicism, X inactivation, some of the words that we threw around quite a bit yesterday. I just want to kind of make sure that we're all on the same page and are aware of what those terms mean and why we talk about them. So going a little bit back, so just a quick genetics overview. So when we think about our DNA is just the information that's inherited, that's passed through families. And I like to think of our DNA as kind of the instruction manuals for how our bodies grow, develop, and function. And our DNA really provides the code to make a protein. And the protein is the working unit within the cell. It's the part of the cell that really does the job. Okay? So we looked back. So this is more of a cartoon. So our DNA are just on long strings. So I like to think of our each individual gene. So we have about 20,000 individual genes, and they're all strung together on what we call chromosomes. I like to think of them as like kind of popcorn on a string. So if we have a string of popcorn, and each little popcorn is a gene, okay? And they're all packaged into these chromosomes that are here. So that's a cartoon, but if we actually were to look at a cell, break open a cell and stain the DNA, this is what all of our chromosomes would look like. So each one of us has 46 individual chromosomes arranged in 23 pairs. So 1 chromosome in each pair we inherit from mom, the other we inherit from dad. When we have children of our own, we either pass on one or the other through families, to our children. So chromosome pairs 1 through 22 are the same, so the genes that are on one chromosome are the same as the genes that are on the other chromosome, so that we have 2 copies of every gene. So the last chromosome pair down here, so females have 2 X chromosomes, and males have an X and a Y chromosome. We call those the sex chromosomes. So because females have 2 X's, we see that the X chromosome is significantly larger than the Y chromosome over here. It contains different genes. There's different information on those genes. So because females have this extra copy, in every cell of the body, one of those copies of the X chromosome gets completely shut off, so that for each cell, only one X chromosome is expressed. Okay? In general, that's just a random process. So it's a 50-50 shot. One chromosome gets shut off, or the other chromosome gets shut off, in most cases. So that's kind of what we were talking about yesterday when we were talking about X inactivation. So one of the X inactivation is the process of just one of those chromosomes being shut off. And because one of those chromosomes gets shut off, females are just what we call natural mosaics. So there's just a random pattern of one chromosome being expressed or the other chromosome being expressed. And that's kind of what is made, that's what makes PCDH19, the inheritance pattern of PCDH19, so unique and so interesting, because it's a combination of being on the X chromosome and because of the inheritance patterns of X genes on the X chromosome, and also the function of the PCDH19 protein, because it's on the outside of the cell, you know, and needing to interact with other cells, and the stickiness of the PCDH19 protein. It's those two factors that have made this particular condition so difficult to study and to understand. Okay. So going back to our cartoon, so I mentioned that our genes are just long strings of letters that provide a code to make our protein. So in general, so every 3 letters of our gene code for one building block of our protein, so one amino acid. And here's another way to look at it. So if this is our long gene right here with our long string of letters, every 3 letters code for an amino acid. So if we have CAT, that's going to code for one particular amino acid, a histidine. So when we do genetic testing, we're really just doing a spell check of our DNA and of our genes. So we say, okay, going through, we're supposed to have a CAT here. Do we have a CAT here? And if we look, if for some reason our A is switched to a T, that completely changes our DNA code, and that could have an effect on the protein because a different amino acid is coded into the protein. Okay? And those particular changes are different for everyone, and that's what we're trying to collect in the registry. We're trying to figure out who has a spelling change, what that spelling change is, and what's the result effect on the particular on the protein. So for every child, everyone has a different spelling change in their DNA, and we need to say, okay, who has what spelling change, and what's the predicted effect on the protein? And that can be sometimes, you know, why. As clinicians, you know, we get a genetic test report, we look at a spelling change, and sometimes it's hard to say with 100% certainty, oh, this is a diagnosis for you, that your child has PCDH19-related epilepsy because of this spelling change in the DNA. Because not all spelling changes in a gene can significantly alter the way that that protein works. We talked a lot about yesterday, you know, looking at the functional consequences of particular variants, and not all variants change the function, and not all alterations of function will ultimately lead to seizures and some of the developmental problems that we're seeing these days. So this is just kind of an overview of, this might be something that you see on your genetic test report, and this is just showing that at DNA position 695, there's supposed to be an A, but there's a misspelling and it's actually a G. And that causes this particular change in the protein where you're supposed to have an asparagine at position 232, but there's actually a serine. So that's just how we kind of document the different changes in the PCDH19 protein. I think I might skip over this for the sake of time, but there are just goes to show you that there are different types of spelling changes that can lead to different effects on the protein. So for example, we talked a little bit about the missense variant, in which case you just switch out the particular amino acid in the protein, but there are other changes that actually tell the protein to stop being made prematurely, so you have a shorter protein than what you're supposed to have. Some people have either deletions or insertions, so that a letter is actually either added or removed from the DNA sequence. And as a result, if you add a letter, so down here, so if you have your CAT, C, and T coding for your histidines, but if for some reason you take out an A, it's going to slide the whole sequence over, and as a result, your protein is going to have a different code from that point on, because your whole DNA code is being disrupted. And something similar for an insertion. So I kind of went on a little bit of a tangent there, but I thought it was important just to kind of touch on why we look at particular variants and why they're kind of important to see different variants. Because I know when I talk to a lot of families, they say, oh, I have the PCDH19 gene, but it's a little bit more complicated than that. We really need to tease out what specifically is going on with the PCDH19 in your child to really pull that together. And I think one of the added benefits of having the registry is just kind of having the opportunity to pool as many variants as possible together to see what's been reported. And I think it's going to have a couple benefits. You know, we're going to have the pool of variants to help researchers to tap into. If researchers want to say we want to look into the functional consequences of particular variants, we have a list of variants that have been reported. And I think also too for clinicians and diagnostic labs having the opportunity to look at variants that have been reported in helping to diagnose kids in the future. So when we look at a genetic test report, when we first get it, one of the first questions we ask is, has this particular variant ever been reported in a child who has PCDH19-related epilepsy? And having databases to look into to collect that information is very helpful when we're trying to communicate that to our families and to provide diagnoses for our families. So just an overview of the PCDH19 variants that have been reported in the registry to date. Most of them about half of them have been those missense variants in which just one amino acid has been changed. And then about a quarter of them have been what we call nonsense variants. So those proteins that have been just shortened, they stopped the protein from being made earlier than it's supposed to. We had a couple of small deletions and insertions. And then a couple of people, 3 of them have had whole gene deletions, so rather than an actual spelling change within the protein, there was actually a chunk of chromosome X that has been missing, so that chunk has included the PCDH19 gene, so it's a complete absence of a PCDH19 gene on that one copy of the chromosome. So that's what we're seeing so far in the registry. And I'm going to step aside and let Nilika talk a little bit more about the clinical information that we're seeing.
Thank you very much. So I wanted to report to our families what we've learned so far from those enrolled in the registry, the clinical aspects, so the symptoms that you're seeing at home in your kids. So in terms of seizure types that are reported, we talked about this yesterday. In terms of what families are reporting, focal seizures are seen in over half the kids, and apparently generalized. So that could be what looks like a generalized seizure but starts in a focal manner, are reported in most of the kids in the registry. The age of seizure onset on average is within the first year of life, around 11 months, but the range can be anywhere from as young as 4 months up to over 3 years of age. So it gives us a sense of when PCDH19 related epilepsy tends to present. Seizures sensitive to fever have occurred in about almost 3/4 of people. And as you can see, the vast majority of kids enrolled are having seizures that occur in clusters. So there are a couple of kids enrolled in the registry where they're not having clusters of seizures, but those are the minority. I wanted to talk about development and cognition. So when you look on the internet about this condition, you'll see reports of developmental delay and intellectual disability, but as we've touched on this weekend, there is a great deal of variation from person to person in affected individuals. So what are the specifics in terms of developmental delay that we're seeing in kids enrolled in the registry so far, and to what degree of intellectual disability are we seeing? To tease apart the developmental aspect a little bit, a few families have seen delay in development prior to seizure onset, so 4 out of 22, and 7 out of 22, so about 1 in 3, have seen a regression or a loss of milestones once the condition has presented. And then to tease that apart even further, about half have seen delays in all domains, so language as well as motor, gross motor, and fine motor, otherwise known as global developmental delay. And then about 1 in a little less than a quarter have not reported concerns in development. In terms of cognition, this is the theme for the weekend, there's such variability. So this pie chart is a good visual representation of the great deal of heterogeneity that we're seeing with PCDH19 related epilepsy. So either mild, moderate, or not applicable delays or changes in cognition. So it's really a spectrum.
What does not applicable mean?
Sorry?
Sorry to interrupt you, but like typical versus not applicable, what do you mean by that?
Go ahead.
Sorry. So typically it's more of a not applicable is people who are really too young. So it really is a converting around age, like about 3 or 4, they talk about developmental delay versus intellectual disability, and kids are just really too young to really say one way or the other.
Thank you.
And then to tease apart the behavior, so people will talk about behavioral concerns, but what does that mean? Is that dysregulated behavior, features of autism, sleeping difficulties? What sorts of behavioral problems are we seeing? So when we talk about behavioral dysregulation on the registry enrollment packet, you'll see particular symptoms. So stubbornness, hitting, kicking, things that need to be done her way. So just what aspects of behavioral dysregulation are we seeing? Tantrums. So all of these things together are occurring in 18 out of the 22 kids enrolled. But we hope with more numbers and we can tease apart individual, to tease that 18 apart even further, to understand which of these symptoms are occurring and potentially even connecting it to the genotype to see which gene change is resulting in which aspects of behavioral changes. And then a little over half, so 13 of the kids enrolled, are not showing autistic features in the registry so far. And a minority of kids, so 4 so far, actually have a firm diagnosis of autism. Sleeping difficulties are reported in lots of kids, so over half. And they run the range from either difficulty falling asleep or sleeping during the day, but staying asleep tends to be the most frequent of the difficulties in sleep that we're seeing so far.
So kind of reflections on the past 2 years. So what we can see is that our current enrollment numbers are relatively small. We know that there are a lot more people out there than the 24 that we have at Boston and the 4 that we have at UCSF. So looking back, we're kind of trying to think about what's working, what's not working, what we can do differently. But just looking and thinking about our enrollment numbers being small, we understand that life is busy and our enrollment process is really cumbersome. It's kind of hard to get to paperwork and getting in touch with us and reviewing the paperwork and signing it and sending it back, and that questionnaire is so lengthy. We understand that. And then on our side, the clinical data collection thus far has been really challenging. The family questionnaire we mentioned is long and complicated, and then we're actually trying to collect medical records. It's very, very time-consuming and difficult. People are very apprehensive about giving us records, and sometimes we're sending in requests to hospitals and clinicians multiple times, and people just aren't sending us information back. So we're trying to think about ways to streamline the process. So we have the things that we identified as areas that we can work on our end. So we know the enrollment process is difficult, and unfortunately it's not something we can change. I tried. I have been contacting our IRB, so our institutional review board is the kind of like the ethics committee of the hospital that really determines how you carry out research studies. And while the IRB allows us to have alterations of informed consent, so those other registries that you can just sign up for online, click a couple buttons, I asked our IRB, I said, is this something that we can do? For example, if we just give you information by clicking this box, you agree and consent to participate. Unfortunately, they don't allow us to do that, and that actually is because we're collecting, identifying information, we're collecting the names of the participants with the intention of collecting additional information long term, and also as a way to recontact families to inform you guys about research studies, clinical trials, things like that. It then becomes a HIPAA issue, so we actually have to have documented written informed consent in order to collect that information. So I tried to bypass that, but unfortunately we couldn't do that. And we're thinking, rather than doing a questionnaire that you fill out, to actually do a phone interview that might be a little bit easier on you guys. And I think too, it might allow us to collect a little bit more, a little bit different information than we can, rather than getting information from you that you fill out on paper and then collecting medical records. Maybe if we do a phone interview that kind of mimics what your neurologist might ask you in clinic, we might be able to streamline the way that we get clinical information. But of course, we'd love to hear your feedback and hear from you guys what's working, what's not working, and ways that you think that might be easier for you. I know that you guys all talk. I've talked to Julie a couple of times in ways that we might be able to streamline this, but we'd love to hear your thoughts. And along the lines of a registry, really the goal of a registry is to kind of have a huge database, as many people as possible. And we know that these efforts are kind of being duplicated in other areas. Nicola yesterday mentioned that he had a very large cohort, and he described his clinical cohort very well. So we've been in talks with them about having a joint international registry. Rather than repeating the same efforts, can we kind of put all of our efforts together and have one large cohort, which all sites kind of continue to collect our own data, and then we just submit it into more of a centralized place. And our hope too is to create an online database that is de-identified, so it's obviously not going to have your identifying information, but it is going to have a little bit of information on the variant, the number of times the variant has been reported, and some very basic clinical information that can be accessible to researchers, which is initially why the registry was started. But I think too it's important to make this information accessible to clinicians and diagnostic labs, the people who are trying to make these diagnoses for other people. The more information that they have, it's going to be helpful for other families as they go through this process as well. So with that, I'd like to just thank, so on the left here is our epilepsy genetics program at Boston Children's Hospital. This was taken on Purple Day this past year. And of course, for you guys for enrolling your children and yourselves into our registry. It wouldn't be possible without you. And with that, we'll take some questions if we have time.
I should also add that for folks who are interested, we'll have enrollment paperwork available at the end of the session today.
So I don't have a question. I just have to get on my little soapbox for a minute. In our PCDH19 group, we complain a lot that doctors don't know how to treat our daughters. Or one doctor says, oh, give this medication, and then someone else is like, that's crazy. Our doctor says that doesn't work. And so it's very frustrating, but collectively we share information, and we just get frustrated that doctors don't talk to each other. But this is the way for that to happen, because nobody knows what our daughters go through unless we have data. And so if we could do anything for our girls or for the future of what happens, they have to have data. So I went through this process, And it's really not bad. Like you guys are making it like, oh, it's this bad process. No, it's a questionnaire, and it's easy to answer because you know your kids. It's like, what kind of seizures do they have? Oh, well, this kind, this kind, this kind. When, you know, how old were they when they had their first seizure? It's easy to answer, and so when you look on the internet and you see these studies and you see, like, we had 8 people and 5 of them were like this, and you're like, well, my kid's not like that. It's because we need more people. So please do this, everyone. Please, 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 please. That's all I have to say.
I couldn't agree more, because you want to have a goal, to have a cure, then you're going to have to do the work to put the information in so people can get data. But I think that you said that maybe someone, you know, you guys to talk on the phone, because I know for me and Lacey hears from me every now and then. Where is that?
Feedback with the speaker.
And so that could be a definite option, because some parents may have smaller children or something, and so that could be an option. So that's a definite, I think, would be a good idea, because you're going to usually talk to somebody anyway. Unless I'm just the rare weirdo that just has to talk to everybody. The one part of the deletion, that's where I get kind of confused. And I got excited when I saw that numbery thing. Is that on our reports?
Yeah.
The letters?
Yeah. So each one of you, so you should get, if your clinicians give it to you, your actual report that outlines the specific variant.
It's the D121H.
Yeah. So it should outline the position in the DNA, the number in the letter change, and the result effect on the protein.
But you had like the CAT.
Mm-hmm.
So that's
not usually on the report. This is usually what you'll see here.
But you had something with histamine, and I'm on a histamine kick. What is that? Is a D121H? Is that something related to that?
So it's a little bit different. So it's histidine is the amino acid. Histamine is something that I think you're referring to.
But we are having amino acid problems too that we've been looking into on the functional medicine side of it. So I'll pick your brain later.
Okay, so I'm one of those who hasn't quite finished things. So my only feedback to you is it's simple and easy if you have a young child. But if your child is in their teens and you have a whole lot of history you've forgotten, and they may be in notes, but I don't know when she sat up or developmental milestones. How detailed do you really need this detail? Or I'm a perfectionist and I want you to have good data, but to go through all my records which are not organized at all, and to find that is taking me a long time, which I don't have. That's the problem for me.
And I mean, it's really, obviously, as you said, you know, the more information the better, but if we have a general sense of was it early, late, or on time, just those are really enough, and kind of, as much as you can break it down, great, but again,
try. She's an only child, so I don't know if it was later on time. But I'll do my best.
We're so appreciative of the time and effort that the families are taking. We wanted a richer database than just a list of genetic changes, because we want to be able to correlate which gene change can be correlated with which symptom, which medication works for which gene change, et cetera, et cetera. So again, thanks so much for taking the energy to do this, and as much information as you can give, but if you can't remember, then that's perfectly fine, and just write that down, and we can maybe get some more information from the medical record if we're able to.
So this is my note to you guys who are just getting diagnosed with younger kids. Do it now while you can remember all the details. Don't wait until they're a teenager.
Well, along those lines, are there any plans to repeat the questions at a periodic basis, say annually, so we can follow what happens with sleep behavior, seizure evolution?
Our plan right now is to do follow-ups every 2 years to see how things progress over time. The questionnaires and the follow-ups will be less lengthy. I promise.
No, you go. No, you go.
Okay. This might be asking way too much, but are you able to look at the data and see any sort of genotype or phenotype at this point? In terms of seizure clusters or intervals or cognition or anything like that?
Kind of consistent with what's been reported. There really just hasn't been a clear genotype-phenotype correlation. We have for example, there are 3 people with whole gene deletions that their symptoms were really different. We have 2 people with the same exact missense change, and they're on complete opposite ends of the spectrum. We're really not seeing that. I think we have small numbers, and I think just the complexity of the type of variant itself and the mechanism of the disorder and how complicated that is, I think it's likely going to go much deeper than the genotype to phenotype correlation that we so nicely see with other genetic conditions.
That's very nice work. I have one comment and maybe one question. I would like just to reinforce the feeling from some of the parents. It's very important to build this resource. I look at it from the perspective that what we see published in scientific literature, which many your doctors actually read or get to read or get to find, it's to some extent skewed because the scientific literature would only report something different. Wouldn't report a standard PCDH19 patient. The standard PCDH19 patient is going to be recorded here. We already see that you have so many carriers which are not affected. We heard from Nilika. So really the question is now, so what is the penetrance of this disorder? And it's hugely important for genetic counseling. You want to know it. All of you want to know it. So it's very important to build this resource, not just from the published cases, but really collect the cases as much as possible, because that's valuable information which you all of you are going to benefit from. And many colleagues who really struggle probably see to 1, 2 patients, then they go into literature, then see these very different phenotype or slightly different because it's unusual patient, because that's the only way how the patient got published. So it's very important. Even though it takes a little bit of extra work, I would very strongly encourage it. My question is that there are diagnostic laboratories, like we had the Tina Diagnostics here, who are doing routine diagnostics. There are other, in US, probably many of them. Have you tried to somehow connect with them to see whether you can get through them to some of these patients, families to recruit, or that's not allowed? Or because it seems to be that, yeah, the recruitment process your registry is based on 2 sites. But can you somehow spread the net, even through American Society of Human Genetics, to advertise the registry so people actually know and can contribute? Actually, the question is proactive.
So we're in the works of doing that. So we've tried to recruit through the National Society of Genetic Counselors, because I think that a lot of times genetic counselors are the ones seeing these variants. One of the diagnostic labs has actually reached out to us for information from us about a particular variant that they wanted to point out, and vice versa. They said, oh, sometimes when we do research studies, if we have a particular variant and there's an ongoing research study, we stick a flyer at the end of the report for clinicians to give to the patients, and we're considering doing that as well. So it's kind of other recruitment and kind of going back and forth.
Yes, I think that the collecting of data perhaps should involve more physicians and more centers, in the sense that you are only 2 centers that are San Francisco and Boston. And perhaps there are some families, some parents that are afraid to put information on the website or other things, I don't know. And they need the support of a physician. So I think that the best thing is to involve the other center of USA. And because perhaps our experience was that we collected a big number of patients because we contacted all centers or the physician, and they send us the information of their patients. So my suggestion is to involve the other centers of the USA, and if we want to do an international registry, perhaps it's better to involve the other one. So.
Hi, thank you. As a child adolescent psychiatrist, I was really happy to see you were actually collecting behavioral data also, because that's great, and you're thinking about it proactively. And I guess as I'm thinking, if you're going to collect it over time, it seems a lot of the categories you have in there are things that you would pick up early in life, so autism or some of the behavior dysregulation. But if you're going to look at this across sort of the life spectrum, are we thinking of more categories? Is that something you'd have in follow-up questions, or you're not asking up front? Or I'm just curious about sort of how you're thinking about that.
I think that, so this was just the first pass in terms of what had been reported in the major categories of symptoms that were reported is what our questionnaire was built on, but I would welcome any feedback about any additional categories that might be included on follow-up questionnaire, or what we should be thinking about. And I was actually surprised to see how common sleeping issues are, and how frequent that we were seeing even in the small numbers of kids that we're sampling so far. So any other suggestions are welcome. But we can go through our follow-up questionnaire to see what your feedback is about that.
The other thing I would think that's important is if you could do some sort of standardized developmental testing at some point in time, because this is anecdotal from the families, and it's a good place to start. But if you wanted to do some longitudinal follow-up, if there was a mechanism to get some kind of funding for that, that could be done locally through, providers in the area, as long as the testing was standardized.
And I think too, one of the purposes of the registry is to kind of more so get some general information and say, these are the numbers that we're seeing, and hopefully, people who are more experienced in doing those types of standardized questionnaires can tap into the registry as this patient population, say, I want to specifically look at this, and be able to initiate another study with this patient population. Is kind of one of the things that we're kind of going towards.
So I'm going to be talking about this next, about what we're planning to do with the behavioral planning and what we have done so far. But I think that's a very good point in terms of getting the standardized testing. And I think it's an even more important point to look at the further categories. But I just wanted to let you know we'll talk about some more details on that in the coming few minutes, I think, or after the break.
Yeah, so one more question, then you just introduced the break, so it's perfect. Now I'm feeling like Phil Donahue here walking around.
So I'm just brainstorming right now, but I'm thinking about, we've been going through this for about 2 years, and I feel like just the small group of women, moms, who we're literally sitting around going, does your kid do this? Does your kid do this? Does your kid do this? And we're all going, yeah, oh, yeah. But those are questions that no one ever asks. I'm thinking, is there an opportunity where that could ever be, is there an opportunity where we could actually come together as moms somehow, and that could be documented? Because realistically, I feel like there's so much that you guys could learn from just what we go through in an everyday life. Because I look at goofy little things that I think, that's kind of OCD that my daughter does this. She's 2.5. She's totally OCD. But when she's doing the exact same thing as her daughter, and you think that is a random, and then you go, oh wait, your daughter does the exact same thing, and you think that is just a weird, they have these exact same tics. It's just coming together, like all of a sudden I don't feel crazy. These little tiny things that are just a part of our everyday life, and you think, oh my gosh, no one does this. Except, but that's what we need, because realistically, that I've learned so much. I feel so much more confident as a mom of going home and advocating for her, and I mean just fighting for these different things because of the things I've learned just from these women. Yes, she can add to this.
It's hard to pick up on little nuances. So kind of when we created the initial questionnaire, we're kind of anticipating that a little bit, which is why you see those blank boxes like, tell us about your child, and then we get like paragraphs. So we tried to get those little nuances, those things that we're not specifically asking, but volunteer these kind of little nuances that your child has, and you might be able to see those patterns.
Yeah,
but you might not know whether to say it or not until you get with other people.
I feel like as we all get to go doing it together, like it just pulls it comes out of us.
Yeah.
Like things that we don't even think about.
The last couple days, like we've all talked about, we had a conversation the other night, and I said Remy had phosphatidylcholine, and she was a demon child. And then she chimed in, she's like, oh my gosh, my child was like that. She was scratching me, biting me. And then another person said, mine too. And so it's like things like that. It's like, I don't think that drug is supposed to do that, but it's very similar amongst us girls. And then I also heard someone say after a seizure cluster, it's like, the behavior's better, everything's good. With Remy, if she is smiling and hungry, I'm like, the cluster's done. She's happy. It's done. I'm not worried. I know exactly when it's done. But when it's like coming up to a seizure and she is getting kind of like she's not listening and we're bracing ourselves, we're like, it's coming. We know it's coming. So there are a lot of similarities in the girls that I guess that wouldn't be on a questionnaire like that. So
I think how we can maybe, you guys can help us hone that in, is can you hook out of the 33 people that are current and whoever ever comes back in or is coming in, we're going to say they're all going to do their work. Can you hook the people that have the same variants up so we can hone in and pick each other's brain? I know there could be some HIPAA things, but if they agree, there could be somewhere in that paperwork, if you would be willing to reach out to other parents that have the same variant, because I think that's how you're going to really hone in similarities to the same people, because we're all like interviewing how old's your kid, what you're doing. And so really it's what you guys are looking at is each variant. So could you pair us up by the variant of who is participating? So as an option, some parents may not want to. I can't imagine they wouldn't. But as an option, because there could be some legal, I'm not sure how that works, but you're in a study. You want to know. And the variants are going to be the biggest thing for us to be able to pick each other's brain to see if there's similarities with each other. Just saying.
I guess one, and then we'll maybe close with this. I guess one potential problem with that is we want the observations to be independent, right? Yes, you guys all talk anyway. And so the paragraphs, I'm sure, are helpful, but I guess my plug would be to try to focus in on what so if you say they do this odd tick, because trying to sift through, and I haven't done this, you guys have done it, pages of dialogue to try and say, well, what is this, what is she actually worried about, right? So try to keep it to the point, because then it's going to have more power if we can do it in a blinded fashion. If we stumble on something where a variant shows that person may have more OCD tendencies, that's going to carry a lot more power, because as soon as you guys all start talking, which is great, but then it's going to get all lost in the wash, right? Because then you'll say, oh yeah, she does that too, and then that kind of taints, I think, the predictive value a bit. So with that, anything else? 10-minute break, and we'll start the last two sessions. Thank you.
Part of it is